Let us bring in, we are very privileged in this country, as you know, to have one of the leading global researchers in this space, not only researching, but also treating. You will be aware of her. If you're not a patient of her, you will no doubt have heard of her. She is incredibly generous with her time to us in the IMNDA, and I am delighted to hopefully able to bring in Professor Orla Hardiman. Orla, I know you were watching earlier. Can you hear us? Clearly not. We can see your screen, Orla, but you may be muted. Are you there?
Can you hear me now?
We can hear you now. This is like a broadcasting dream for me, where you're talking and no one can hear you. Orla, we can see your screen and we can hear you. Delighted to have you with us. It's over to you. I will give you a little bit of a nudge with about 5 minutes to go, if that's all right.
Thank you very much. And actually, it was really interesting listening to Nick and Richard. But actually, we do have voice banking in Ireland. And if people want to discuss about voice banking in Ireland, it is available through our national clinic in Beaumont. So obviously, those apps are really interesting from the UK. But we have services in Ireland as well. So we're happy to discuss that with anybody. I'm going to talk about research this morning and why I think we should be really optimistic about what's going on with motor neuron disease research. So we know that motor neuron disease is a probably gene-environment interaction, and we know from the news that was going on in the last few days about rugby players that one of the environmental exposures may be head trauma in some instances, but that probably happens in the context of somebody who's already got a genetic predisposition, and that genetic predisposition might also make you very good at sport. We also know that there are certain genes, and we know in Ireland that the gene called C9orf72 is a gene that's important in motor neuron disease, but having the gene doesn't necessarily mean you're going to get the disease, but it does reduce the number of events that you need to get the disease. So we know that it's a gene-environment interaction. One of the pieces of research that's been going on, which we've been involved for many years, is to look at the genes that might be associated with motor neuron disease. And there are about 30 at the moment. But in Ireland, really, there's only one that's very important. It's called C9orf72. There's another smaller gene, which isn't listed here, called ataxin-2. And there's a gene called FUS. We have a few people with FUS over the years in Ireland, and they're important genes because I'm going to talk about them a little bit at the end. The other gene that's important is SOD1, and that's an important gene because that's been in the news very recently as well and has caused great optimism. But unfortunately, fortunately in another way, SOD1 is not very important in Ireland. We've never identified anybody with familial SOD1 in Ireland. We identified one boy about 20 years ago who had an SOD1 variant, but it wasn't a familial form. So it's not an important gene in Ireland. So I'm going to talk about that a little bit later. But there's been some developments there that make it very exciting. So these are the genes that are important, but really in Ireland, the most important one is C9orf72. Now, what we also know is that lots of laboratory work that goes on, a lot of publicity around work that goes on in the laboratory, these breakthroughs that happen in motor neuron disease regularly, but they're nearly always in animal models. And the animal models are very useful because they tell us a lot about the biology of what's going on, but they don't tell us about the disease in its entirety as it happens in people like the people in the room here. And the reason why is because the human brain is much more complicated than any of the models that we use. So the models are very valuable because they tell us a bit about the science, the basic biology, what's happening, and we can just dissect out different types of biological mechanisms that are probably important in the disease in the fly or in the mouse or in the laboratory cells. But translating that into what's going on in the human brain is much more complicated and is problematic and has led to difficulties in terms of breakthroughs that happen at a laboratory level and then translating that into what's happening at the human level. And one of the things that we realize now is that, and it came up with a question earlier, is motor neuron disease like cancer? In other words, are there more than one forms of motor neuron disease? And the answer is yes. And that the way we need to approach treating motor neuron disease is the way we now approach treatment of cancer, which is the idea that people have an individual manifestation of the disease. We can probably segregate that manifestation into subgroups of people, and precision medicine is a way to do that. It's a way by which we tailor these medical treatments to the individual characteristics and our ability to classify people into subgroups that differ in their susceptibility to the disease, both in their biological process and the way the disease progresses. And that's quite well advanced in cancer, but it's more difficult in motor neuron disease. But we are beginning to be able to dissect out the genetic factors and possibly environmental exposure factors, the factors that then lead to the disease, and then the factors that drive progression. And they all may be different. The factors that lead to disease risk may be different from the factors that lead to disease progression. That's one of the big sort of unknowns and unknowables at the moment. We have some genetic data that suggests that they're different, that the risk factors in terms of developing the disease may differ from the factors that lead to the disease progression. And that's very important in terms of how we target specific treatments for people. We know, as I said, I've seen maybe 3,000 people over the years with motor neuron disease. We were just looking at our register this morning. And we know that we can, if we use different methodologies, we can subdivide people into different subgroups. We can do that clinically to some extent. And people often ask us, well, what type of motor neuron disease do I have? And we say, well, it starts in your legs, your arms, so you have spinal onset, or it starts in your speech, your swallow. So it's bulbar onset, or you have some thinking trouble, or you don't have thinking trouble. So we have some ways of segregating out, but there are a lot of other biological or technological mechanisms that we can use now that will help us to subcategorize people. Genetics is one of those technologies that we're using to do that, and I'll talk about that a little bit towards the end. Our mission, and the mission that I have and that I have with my colleagues across Europe, is to find the right drug for the right person in the right dose at the right time. That's the mission of Precision ALS, Precision Medicine. It's a mission of the TRICALS, the Treatment Initiative to Cure ALS, our European clinical trials group. And it's a mission that we have on this new initiative that we set up recently called Precision ALS, this way of building a precision medicine approach towards treating motor neuron disease. I'm going to talk for the rest of my talk here about clinical trials because it's very much in the news at the moment. And we often get asked, well, you're putting me on Riluzole, and Riluzole has been around for the best part of 30 years. What are you guys doing? It's really disgraceful that you haven't found a treatment that works, and this drug that you're using has been around for so long. It's a really old drug. And I have a lot of sympathy for that view. It's not that we haven't been trying, though. There's been many, many trials of many different drugs, and probably more like 50 or 60 now. Hundreds of millions of euros have been spent on drug development and on bringing drugs to clinical trial and on identifying whether the drug works or not. And uniformly, with some notable exceptions we'll talk about later, these have been negative. So there's something called the valley of death, where the basic scientists produce drugs that look like they're really effective in the cell or in animal models, and they bring it over to us in the clinic. We do a clinical trial, and the drug fails spectacularly. And there are lots of reasons why that has been. The animal-based approach hasn't been very successful. The effective treatments are more likely to be effective in subgroups of patients. We also need better markers of the disease, better markers to subcategorize the disease, and better markers of whether the drug is actually doing what it's supposed to be doing, and better markers of whether the disease is progressing or not. We also have had problems over the years in the way we design our trials and the way we measure whether the drug is working or not. And there's a lot of research going on in these areas. I suppose the most spectacular failure was about 10 years ago, a drug called dexpramipexole. We did the trial here in Ireland as well. Everybody assumed the drug was going to work. The phase 2 trial, which is an early phase trial of a smaller number of patients, looked really positive. And then when we did the larger phase 3 trial with about 800 patients, there was no difference between the people on the drug and the people that weren't on the drug. And that led to a lot of sort of soul searching, rethinking about where are we going wrong here? The phase 2 looked really good. Phase 3 was negative. The drug died, didn't work. Why did we think it worked, and where did we go wrong in bringing that drug to an unsuccessful conclusion? Now, that's the past. What about the present? Well, the present looks a lot more hopeful. We've been getting a lot of questions about tofersen. So tofersen is a gene therapy, and it's a gene therapy for the gene SOD1 that I mentioned at the very beginning. And we know that SOD1 was the first gene to be identified in motor neuron disease, and it accounts for about, it depends on how you look at it, but about 1% of motor neuron disease in populations European extraction. It accounts for maybe 15% of familial motor neuron disease in the United States. But the frequency of SOD1 mutations in Europe is quite variable. We don't have any in Ireland. There aren't any in Belgium. There are some SOD families in the UK and in France and in Germany and in Belgium. The point about this is that the Biogen and Ionis, which are two companies, developed a gene therapy for the abnormal gene in SOD1, and it's a little strand of genetic material that you inject into the spinal fluid of people with the SOD1 related motor neuron disease, and then determine whether that was effective or not. And it looks like that works to some extent. It looks like it slows down the disease. So tofersen, this was published just very recently, a couple of weeks ago, and it's created a huge stir around because it's the first treatment that looks like it might be beneficial. It hasn't got FDA approval yet. Actually, the trial wasn't actually technically successful. The trial was a bit short and there were some difficulties in the trial design, but when you look at the subsequent data, it's really positive, and there are lots of other markers that suggest that the drug really was working in this particular subtype, but we don't have this subtype in Ireland. But it's a really important breakthrough because it shows that gene therapy for certain genes may be beneficial. The other drug that we've been asked a lot about is this AMX0035. That's the Amylyx study. This is so we're doing this study in Ireland. The study is called Phoenix. It's a study of this drug AMX0035. It's a placebo-controlled study across a number of sites in Europe and the US with two drugs called sodium phenylbutyrate and taurusidol, also Trutka, in people with ALS. And this trial is underway. But Amylyx, on the basis of a phase 2 study with a very small number of patients, submitted to the FDA. And last week, the FDA approved the Amylyx 0035 for use in the US, and it's being marketed as Reliviro. Now, this caused a lot of concern and controversy, this approval. I was actually at a meeting in Amsterdam last week, a workshop with all my colleagues and a lot of industry partners, and this approval we sort of anticipated because the FDA has been approving drugs quite early for motor neuron disease. They approved edaravone as well, but it raised some questions, and actually it also raised some questions within the business literature as well. So this is a news feed that came out. I want to point to the study was only 137 patients, and I want to remind you that the study that we did at dexpramipexole was a slightly large number of patients, a phase 2, which looked like it was positive, and we went and did a larger study called a phase 3 study, and that study was negative. And here's another news feed from the Business and Finance, I think it was. Drug is going to cost $158,000. So it's not even clear if the payers are going to cover that, and it mightn't even work. We don't know. The phase 2 study, 137, I've seen the data. It's very hard to know whether that drug really works. A very small cohort of patients. So it was a 2-to-1 ratio. So only about 100 patients got the drug. And so we don't know really if that drug works or not. So we did a consensus statement, and we wrote this when I was in Amsterdam last week. So this is the European view on AMX 0035. While it seems to be unfair that people in the US are most likely to be able to get this drug, while those in Europe can only get the drug as part of a clinical trial, we believe there's very good scientific reason to complete the study in Europe. We don't know for definite that this drug works. The US data were limited in time and patient numbers. And if we all took that approach, we would never know whether this drug works. So we remain committed to providing the most effective therapy for all people with this disease. And we will continue to conduct high quality studies that provide definitive results regarding the benefits of people with MND. And I think this statement is on the website, the IMND website. So I'm happy to take questions about this. This drug is also been approved by Health Canada, but I've been speaking to my colleagues in Canada. It's not clear that even Canadian patients will be able to get this drug, because it's not clear that the payers, either in the US or Canada, will cover the cost of this drug. What are we doing in Ireland? Well, these are the studies that we're doing at the moment. We're doing a study on edaravone, which is not available in Europe, but we're doing a clinical trial. Courage is a muscle strength drug. Phoenix is the AMX 0035. WAVE is a gene therapy study. We've just finished a study in Trutka, and the remaining studies are underway. Lighthouse it's going through approval at the moment. That's a drug called Triamec. Magnet is lithium, and the other drugs are smaller drug trials that we're just going through approval. So we have loads of clinical trials underway at the moment. I don't do trials of drugs I don't think are going to work, so I'm very hopeful that we're going to have lots of drugs that are going to work in the near future. What about these? I mentioned about this gene therapy study, the tofersen study. There are other gene therapy studies underway at the moment. There's a gene therapy study for CNRNOF72, the gene that's common in Ireland. About 10% of people in Ireland carry the CNRNOF72 gene. About 50% of people with familial motor neuron disease in Ireland carry this gene variant. There was one study, a gene therapy study that we're involved in, conducted by the company Biogen and Ionis, which was negative for this using gene therapy for this gene. And we're involved in another study now, sponsored by another company called Wave, slightly different design to determine whether there's any benefit. So we're currently enrolling people in very small numbers, 124 people around the world. So we're a site for that. Ataxin-2, we think, is that a modifier gene doesn't actually cause the disease, but it might change the way the disease progresses. So Biogen and Ionis also have a clinical trial running. We're not doing that, but there will be a larger trial in the near future we will probably do. And FUS, which is a really rare mutation, a rare gene, but we're a site worldwide for FUS mutations. So we will do FUS gene therapies, even if they're not Irish patients. We'll accept patients from all around the world with FUS mutations to conduct that study.
About 5 minutes, please,
okay.
Orla.
What else can we do? Well, make more progress by working together. There are better ways of selecting people and better ways of targeting. There are 70 new compounds in clinical trial, but we need to be better at generating our clinical trials. So I want to talk for the last 5 minutes about this new initiative called Precision ALS, which is an Irish-led academic industry collaboration. These are the objectives that we have in Precision ALS. It's really about working with data scientists to build a new patient data platform, collect across the whole of Europe people with ALS or motor neuron disease, put them on this patient data platform. These are all my colleagues in Europe. We're leading out on this. This is the big operational team. It's enormous. A lot of legal stuff, a lot of industry partners, and we're just about to start. This is how the data flow. This is why we're asking about Google, because data flow and data governance and data curation and data privacy is really important. So we're working very hard on this. We're going to be asking people to allow us to collect their information so that we can put it onto this patient data platform. We'll be collecting additional data to try and identify new markers of the disease so we can really generate a very strong precision medicine approach. These are types of data, imaging data, EEG-based data. This is some of the work some people in the audience will have participated in our imaging and our brainwave studies. These are really cool data that we can generate here. And then better outcome measures. We're working with a number of companies. Actually, we're working with a speech company as well called Oral Analytics to look at early changes in speech and breathing. We're working with lots of device companies to map electronically what's happening to people over time. We're working with a company called MyPath in the UK, ADI. We have a system called TIM, Telemedicine Motor Neuron Disease. Some people are participating in that with us as well. And then Miriam is going to talk after us about the patient journey and what we're collecting there. Ultimately, this patient data platform is going to look like this, but ultimately we're going to have better clinical trials so we can identify people who are suitable for different subtypes of drugs and different outcome measures that we can really use the data to drive whether the drug is working or not. So to conclude, we know that motor neuron disease is a complex condition. Different people have different subtypes of the disease. We're beginning to understand the disease complexity. Working together will help to speed things up. And there are new and more effective treatments on the horizon, and we're right in the thick of that, and we're very excited about what's happening. This is the funding that we have, and we're very grateful to the support that the IMNDA continues to provide in our ongoing studies. I should mention that IMNDA, I think, is committed to supporting the Lighthouse study, which is what we call an investigator-led study, not funded by industry, but funded by charitable organisations like the IMNDA. Thanks very much for your attention. I'm happy to take questions. So that's my dog.
Thank you, Orla. I'm not sure whether your dog was adding commentary there or a criticism, one or the other. Orla, thank you again for all the dedication that you put in to supporting people with motor neuron disease. There are inevitably questions in the room, and there are questions from those watching on Zoom as well. So down to the Q&A with the people who are on the Zoom or the chat. In the room, if you'd like to put your hand up, and we will get a microphone to you and we'll ask some questions. Yeah, if you wouldn't mind saying your name first of all, madam.
Hello, my name is Dervla de Puer, and I'd like to thank Professor Hardiman for her really very interesting talk. It's great for those of us with MND to see what's happening. But my question, apologies, my question is, how do we volunteer or put our names forward for the clinical trials, and how are those choices made? I have one more specific question about the gene-specific clinical trials. I'm wondering what genes have been tested for in the cohort here in Ireland regularly or as a matter of course, and are there some genes that might we put ourselves forward to be tested for?
Okay, thank you very much. We might do the genetic question first, Orla, and then I suppose on the selection process for the trials.
They're brilliant questions. Thank you. Hello, Dermot. So in terms of the genes, yes, we offer screening for C9orf72 to anybody who wants it. It's expensive to do, and don't tell my CEO that we do it because he's probably trying to stop me doing it. But we offer, so C9orf is the most important gene in Ireland. We ask everybody who comes to our clinic to give us a DNA sample, and we regularly do a surveillance of the gene variants in Ireland. We're not allowed to give those results back because it's done in a lab by a postdoc, so it's not in accordance with certification. We can do gene panels in the UK as well. It's expensive, so we do them. If somebody doesn't carry C9orf and they're from a family, we'll often do another gene panel. We haven't found any of the other genes that are associated with motor neuron disease yet in Ireland. So we do surveillance, and once the ATAXN2 trial comes in, we'll be looking for that as well. And similarly, the Magnet study is another. The gene is called UNC13A. We'll be doing that as well. But at the moment, we're not enrolling that trial yet. With respect to inclusion criteria for clinical trials, it's a big bone of contention because we've looked at the data. So, for example, the Edaravone study and also the AMREX 003, study, Senator study was the study that FDA approved. If we had been enrolling that study in Ireland, less than 5% of people in Ireland would have been eligible for that study. So that's what I mean, that 95% of people wouldn't have been able to enroll in that study. The enrollment criteria, between 5 and 15% of people meet the enrollment criteria for studies. We are trying to expand that out. We have other ways of trying to manage the enrollment, but basically at the moment the clinical trials are usually you have to be diagnosed for less than 24 months. So you have to have had the condition for less than 24 months. You have to have reasonable breathing, more than about 60 to 65% on your breathing. And you have to, unfortunately, the disease has to be progressing. So then the general rules, each trial is a little bit different. But we have a clinical trial team that works for me in Beaumont, and we're more than happy to discuss with people who want to be considered. We're very happy to talk to people in the clinics about trials if we think people are eligible, and then we'll reach out to people as we begin to enroll in the trials. So more than happy to talk to everyone separately and offline about this.
So that's probably the best way to do that. Alfie's asking a question: does each MND patient know the genetic factors that may be linked to their disease at diagnosis? I'm guessing the answer to that is probably not at this stage, but is that where we want to head to, Orla?
Yeah, I think that's precision medicine at its best, and the answer is that we know the big genes, so we know if people would carry C9orf or not. We're part of a big group called Project Mind, which is a whole genome sequencing project. It's expensive to do whole genome sequences, €1,000 for each genome. So we've done this philanthropically. We got a big donation from a donor, and also Science Foundation Ireland helped us. So we've contributed to Project Mind. Project Mind does do large-scale genome sequencing and looks at patterns of gene complements that are at risk. We're not quite there at an individualized level, but I think that is going to be the next step sometime in the future. We're not quite there yet. There's a lot of maths involved and a lot of statistics involved, but Project Mind is a very big initiative. My colleague Russell McLaughlin, who's a geneticist who did his PhD at me, but now is a grown-up and manages his own team, he's very involved in this, and we work very closely with Russell and with the teams around Europe who are involved around the world now who are involved in Project Mind. And Precision ALS, which is the clinical data collection tool that we're using, which we're driving in Ireland, that'll link tightly with Project Mind. And I think ultimately that is the holy grail, to identify the susceptibility genes that people have and link those in with the type of therapy that they have.
Wonderful. Okay, we have another question in the room if we can. If anyone has a question, they can put their hand up. Yep. And we'll come over here then after that. So again, you might just say your name for Orla, who might not have heard your first question.
Hi, I'm Ruth. Thank you very much, Orla. I just wanted to clarify what you were saying about the genetic markers. You said that when a patient comes to see you, they submit a DNA sample. So do you mean that then that DNA sample is screened for C9orf72 and is SOD1 routinely screened? Because you said that you haven't identified anyone in Ireland with that carrying that gene. I just wondered, how sure are we, that there aren't people walking around with that gene? Yeah.
Okay, good question. So when we see people in the clinic, we offer C9orf for people who are interested in it, which is done in a certified lab in Sheffield. So that's a sample that goes over to Sheffield, and it takes a few months because of COVID. There's a delay in getting that back. We don't routinely screen for the other genes because the gene panels are expensive, and we know from the surveillance work that we've done over the years with the second sample that we asked people for, which is for our DNA bank, we do a screen every now and again, every couple of years, to make sure that we haven't missed out people or within the population variants that we know to be pathogenic. If that were the case, we would go back and rescreen the population. So we do it. We're pretty confident that we haven't seen any SOD1. People who've got familial motor neuron disease, SOD1 is almost always familial. People who've got familial motor neuron disease who don't carry C9, we screen for the other genes as well. So we ask Sheffield to do a second screen if the C9 is negative. So I'm pretty confident that we don't see it. We're not the only country where we don't see it, by the way. There's no SOD1 in the Netherlands either. And outside of North American and European ancestral populations, it's pretty, and in Malaysia, it's present in Malaysia as well, and in Japan. But the South America, for example, Africa, there's very little SOD1, and no C9, by the way. C9 is a Northern European variant as well. So we're pretty confident that we're not missing it, actually, is the answer.
Okay, thank you very much. We had that question from over here, yeah.
MK here. Is there any drugs or trials for PLS patients?
I was getting to your question, Michael. It was on my list to get to. He texted me as well to make sure I ask him. Orla, I'll let you answer it.
That's a brilliant question. And the answer is, at the moment, no, but we're part of a large international consortium. And actually, Peter Bede, who's the medical patron, has done probably the most important work internationally on PLS in the recent past. So there is a consortium led by my colleague Hiroshi Mitsumoto in New York, who's really looking to see what we can do for PLS. The difficulty with PLS is because it progresses very slowly. It's very hard to measure change in a short period of time, and the clinical trials are designed to measure change over a short period of time. In fact, in our opinion, the trials are too short. The trials are 6 months, and they should be longer for typical motor neuron disease. So PLS, it's difficult to do. If we have better markers, and actually in our research work, we have got some markers using brainwave testing, EEG, and something called cortical muscular coherence. So we have some markers that we think will be very useful to see if we can detect change in PLS. So the answer is not now, but it's not that we haven't been thinking about it. It's not that we won't be doing it. It's just it's been more difficult and more challenging because of the rate of progression of the disease. So it's a really good question.
Okay, thank you for that, Michael. We will have the Bishop's Crows. You're dragging me off stage. Tracy Hutchins will give now to me if we keep going too long, but we're going to get as many questions as we can. Mark Dignam asks via Zoom, could Professor Hardiman comment on the follow drugs, and forgive my butchering of their pronunciation, Proleukin, Triumec and Terasozin. So there's three there, Orla. I'm not sure how familiar you are with each of them.
Yeah, I do know about them. Proleukin is an immunomodulatory drug. We're not involved in those trials. We're waiting for the outcomes of a trial called Miracals in the UK, which is looking at the immune modulation. So that should be reporting out in the next few months. Triumec is the Lighthouse study, so we are doing that. And the third one is Trazodone, which is the MND Smart drug. Now, I reviewed that project. It's not a drug we're particularly interested in studying for a number of reasons. The clinical trial design in MND Smart, the Scottish study, I think probably won't give us the readout that we need. So we're focusing on drugs that we think are likely to be beneficial. We're interested in Triumec, and Triumec is the Lighthouse study, and IMNDA is going to help fund that, actually. So we're very excited about that. It's going through regulatory at the moment.
Okay, and one more from Dennis on drugs before we move off it. Is Rilutec still regarded as an effective treatment? That's from Dennis Fitzgerald.
Interestingly enough, we've looked at riluzole and its use of riluzole in people who've been on riluzole and people who haven't. We've looked at it a few times. And actually, I just reviewed some data this morning with Rob McFarlane, who's one of our new fellows, who's just looked at our register again. And the curves of people who are on riluzole are much better than the people who aren't on riluzole. So yes, riluzole is effective. And it may also be beneficial as an add, if we add on the new drugs that we are hoping would be beneficial, they may work synergistically with riluzole. It probably doesn't work the way we thought it worked. It probably has a mechanism of action that isn't really very well understood. But the answer is yes, it does work, and we should be using it.
Okay, Orla, you've been very generous with your time. Ladies and gentlemen, Professor Orla Hardiman. Thank you, Orla, and looking forward to working with you closely over the next few months.